Thank you all for coming to the Wednesday afternoon lecture today, March 21st, 2012. My name is Steve Zula. I'm the Mitochondria Interest Group Coordinator. I'm also a Program Director at National Institute of Biomedical Imaging and Bioengineering. It's my great pleasure to introduce to you not our speaker, but the Scientific Director for NHLBI, Bob Balaban.
Well, thanks and welcome. The first announcement I want to make is that there will be coffee and snacks afterwards, and we should all thank FAES, who has come to the rescue of NIH again. So we greatly appreciate that. It's my pleasure to introduce Dr. DeMario, who is the Lucy G. Moses Professor of Neurology at Columbia University. He received his PhD and early training in neurology at one of the great mitochondrial universities in Europe, and that's Padova. But I understand that was not his real interest at that particular time. I thought that may have been the origins of his interest in mitochondria, but at that time, as you know, people know his career, was really originally in glycolysis. From there, he went on to do a postdoctoral position at the University of Pennsylvania, where he then told me where he really got interested in mitochondria, where probably a lot of people in this room got interested in mitochondria, and that was, if you will, the knee of Britton Chance, who just recently left us not too long ago, but it's great to see his impact still moving through the scientific community. He advanced to assistant professor there, but then in 1974, he then moved to the Neurology Institute at Columbia, where he has been since then, and is a very, very happy New Yorker, though he does spend a few weeks in Italy at a very nice location every summer, so he's not totally abandoned his ties. His research interests have been consistently focused on inborn errors of energy metabolism, including defects in glycogen metabolism, and I know as many of his friends here, in glycogen disorders in fatty acid utilization, and defects in the mitochondrial respiratory chain, and the clinical impact of these defects. And in fact, he just told me he's helping one of my old fellows learn about clinical implications of mitochondrial disease, Vamsi Muthra, which he just recently published a paper with, which I'll mention. These studies led to the discovery of one of the first errors of fatty acid oxidation in humans, and to the identification of new enzyme defects of muscle glycolysis. And again, as I mentioned, most recently in a large team collaboration, he's demonstrated the utility of targeted next-generation sequencing in the evaluation of mitochondrial diseases. And I think this is really a major step with regards to personalized medicine in terms of the mitochondrial field. And it's a very seminal paper in Science Translational Medicine, which I suggest you look at. So I look forward to your talk. We're very interested in hearing about mitochondrial DNA and human disease processes. It's in his talk. The talk entitled is Mitochondria DNA, Prometheus Gifts, or Pandora's Box. So I assume we're talking about the gift of fire from Zeus here.
Exactly. Thank you. Thank you very much for your much too generous words, and thank you all for being here, including many, many friends, old friends. And it's an honor, of course, to give this lecture. And let me start with the title. In Greek mythology, as you know, the giant Prometheus stole fire from the Olympian gods and gave it to the humans. And that was a crime, which the gods punished by tying Prometheus, chaining Prometheus to the Caucasus, where else? Where he was forever, where his liver was forever devoured by a vulture, as you see in this Greek vase. And actually, I think I see there an intuition of the extraordinary regenerative capacity of the liver. But the plot thickened when the gods, not satisfied with this punishment, sent him also, as a punishment, a very beautiful woman, Pandora, excuse the sexism appearing here, who was and endowed her with a vase in In the European tradition, we say Pandora's vase. So here I imagine this could be it, which contain old evils. And Pandora, unfortunately, was, like most women, very curious. And the prohibition was to open the vase. So therefore I see the mitochondrion as Prometheus bound, because in fact the mitochondrion was independent, and it has been for a long time bound into our cells. And this is a beautiful image, scanning microscopy of the mitochondrion. I don't have to give you details, but you see this is the only double membrane organelle in the cell. And you see the crystal, invagination of the inner membrane. And this is, you'd see, another mitochondrion. Now, this has become my logo, but as you all know, it is in fact the Neil Armstrong's footstep on the moon. And of course, as a mitochondriac, I think it's only appropriate that the first message we left in space is that of the mitochondrion. Mitochondrial DNA was discovered back in 1963 by Birgit and Sylvan Nass in a joint paper, in a back-to-back paper, as you see here. This is the real thing, the double-stranded and circular mitochondrion. This is a schematic view, very small molecule, as you know, 16.5 kilobases, a ridiculous amount of DNA compared to nuclear mitochondria, the mitochondrial DNA, to nuclear DNA, but very important, as I hope I'll convince you of. But in fact, the story of mitochondrial DNA is much older than the myth of Prometheus. It goes back about over 1 billion years ago, when, as you all know, protobacteria, which were able of utilizing oxygen metabolically, took permanent residence in early eukaryotic cells that did not have that capacity. And this symbiosis became permanent, and the protobacteria became mitochondria, and the rest is history. And although mitochondria have a multitude of different functions, I think it is fair to say that probably one of the most important, if not the most important, is the production of energy. And in fact, they do produce most of the energy for the cell. And that's why in this very schematic representation of a mitochondrion with its double membrane, 2 membranes, and showing the origin, the fuel that mitochondria utilizes to generate energy, namely carbohydrates, which are mostly utilized in the cytoplasm, and fatty acids, which are mostly utilized within the mitochondrion in the beta oxidation cycle, although the transport is really quite complex. Well, here I'm showing the terminal pathway, the respiratory chain, which is really the energy generating pathway, the business end of energy generation. Okay, here is a closer view of what I wanted to say is that I'm showing this pathway, this respiratory chain pathway, not only because of its metabolic importance, but also because it is the only pathway in the cell where 2 genomes are needed for it to function, the nuclear genome and the mitochondrial genome. And here is a closer view of the relationship between the mitochondrial genome and the respiratory chain. And the genes in the mitochondrial DNA are color-coded to the proteins in the respiratory chain that they encode. Only 13 proteins, very few, but as you see, essential. 7 proteins in complex 1, none in complex 2, keep this in mind for a few minutes, 1 protein in complex 3, cytochrome B, 3 catalytic subunits of complex 4 or cytochrome oxidase or COX, and 2 subunits of the ATP synthetase gene. So a very small amount of proteins. Notice how in the respiratory chain electrons, as you all know, pass horizontally, flow horizontally from one complex to the other, and the energy, the redox energy that is generated in this series of reactions is preserved initially as chemoosmotic potential. And then, in this way, the mitochondria becomes really a capacitor, if you want. And then when energy has to be generated, the chemosmotic potential is generated by the pumping of protons at 3 sites in the respiratory chain, complex 1, complex 3, and complex 4, from the inside to the outside of the inner mitochondrial membrane. And then these same protons flow back into the mitochondrial matrix at the level of complex 5, or ATP synthesis, generating ATP, and as I'll show you in a second. In fact, I cannot resist but showing you for a moment, for a few seconds, this representation of this magnificent, probably the smallest rotary machine in existence, ATP synthesis, which is made of a rotor, as you see here, and a stator. The rotor lies into the endomitochondrial membrane. The stator projects into the matrix. And as protons flow into a channel of this complex, the rotor turns at the respectable speed of about 1,000 RPM. And this movement sets in motion this cam, which opens the slices of the stator, allowing ADP and PI to come into this portion of the ATP synthesis, generating ATP, which is then released into the matrix, as you all know. But this is a phenomenal, and by the way, ATP is needed every day in a huge amount, quantities. So this machine is in constant activity. So let's go on. Okay. If you have a new DNA, you have a new genetics, different from the Mendelian genetics that we all learned in college. And that's mitochondrial genetics, which needs to be known now by practicing physicians if they don't want to make big errors. So mitochondrial genetics differs from nuclear genetics. How? Well, in 3 major ways, as you know. First, maternal inheritance. At fertilization, of course, we receive the same exact amount of genetic information from father and mother, but all mitochondria and all mitochondrial DNA derive from the oocyte. The second important rule is heteroplasmy and the connected concept of the threshold effect. Heteroplasmy means that while, again, for nuclear DNA, we are dealing with 2 alleles of each gene, with mitochondrial DNA, with mitochondria, we are dealing with hundreds or thousands of copies of mitochondria per cell, and even higher number of mitochondrial DNAs, because each mitochondrion contains from 2 to 10 copies of mitochondrial DNA. So, mitochondrial genetics is in fact population genetics, not Mendelian genetics. And for any clinicians in the audience, the first question will be, well, what will be the consequence at the clinical level of that? Well, the consequence will be that if you have a deleterious mutation in mitochondrial DNA, the phenotypic consequence will depend not just on whether that mutation is present, but at what concentration. So it will depend on the relative amount of mutant versus wild type mitochondrial DNA. And there will be a minimum level of mutant mitochondrial DNA which is needed for the respiratory chain not to function properly and for a phenotype to appear. This is called the threshold effect. It is also intuitive for all of you that the threshold effect is a relative concept. Those tissues that depend highly on oxidation will have a lower threshold than those that depend less, depend more on glycolysis, if you want. Finally, the third important difference is mitotic segregation, which is sort of barely illustrated here. Namely, from one generation of cells to the next, the percentage of mutant mitochondrial DNA can change, either increasing or decreasing. And therefore, not only the genotype, but also the phenotype may change in real time in the life of a patient. And this is not just theory, this is something that has been verified in clinical practice, as I may be able to show you if I have time. The other concept that you all know is mitochondria are present in every tissue of the body, no exception, not even the erythrocytes, because of course young erythrocyte, erythroblasts, do contain mitochondria and mitochondrial DNA. So mutation, deleterious mutation in mitochondrial DNA can and often do affect a multiplicity of tissues, many if not all tissues. Again, there are some tissues, post-mitotic tissues and highly oxidative tissues that are more sensitive to mitochondrial DNA mutations. The brain, skeletal muscle, heart muscle, the kidney, the inner ear, and the retina and others. So that immediately provides the clinician with a few red flags, if you want. As I mentioned a moment ago, if you are dealing with a multi-system disorder, you have to think about the possibility of a mitochondrial DNA mutation. That's not obligatory, but you have to think about it, particularly when you have some typical syndromes that I'll show you later, but also in any such situation. I can't see even that far. There are some symptoms that are very common in patients with mitochondrial DNA disorders, such as short stature, hearing loss, diabetes. Maternal inheritance, when you see, is a big clue. But keep in mind that, as you'll see in a moment, because of heteroplasmy, very often maternal inheritance is not apparent. And only the patient will show flagrant symptoms of mitochondrial disorder, while the mother and maternal line may be only mildly or not affected at all. Okay, and the muscle biopsy, I'll come back to that. Now, this is just a view of maternal inheritance, which is that the mothers transmit the mitochondrial DNA mutation to all their progeny, but only women will carry it on to their descendants. And there will be never, and there's never an example of paternal transmission. And now, because maybe of my background as a myologist, I have to spend a few minutes to convince you of the importance of the muscle biopsy for the diagnosis of mitochondrial disorder, and in particular mitochondrial DNA-related disorders. So these are serial sections, as you see, cross-sections from a muscle biopsy of a patient with Kienzler syndrome, I'll come back to that, with a mitochondrial DNA disorder. And there are three stains here. The modified Gomori trichrome stain, which was introduced in the field by King Angle back in the early 60s. The succinate dehydrogenase, which gives a blue color, and the cytochrome oxidase stain, which is a brown, that gives a brown color. Now, you see that some fibers have ragged red appearance here, like this fiber. Namely, they have purplish coloration, usually under the sarcolemma, which simply represent a large accumulation, proliferation of mitochondria. This is seen better with the succinate dehydrogenase stain. Let me remind, I reminded you when I showed you the respiratory chain, the succinate dehydrogenase, or complex 2, is the only one completely encoded by nuclear DNA. So it doesn't give a hoot whether or not mitochondrial DNA are affected. It only shows mitochondria. It's a good marker of mitochondrial mass. And therefore, when you have mitochondrial proliferation, you see these, we call now ragged blue fibers. The cytochrome oxidase, on the other hand, as I told you, contains 3 subunits, indeed the catalytic subunits, encoded by mitochondrial DNA. So whenever there is a deleterious mutation in mitochondrial DNA, you see no lack of cytochrome oxidase activity. The ragged fibers are COX negative. And then my friend Eduardo Bonilla, unfortunately recently disappeared, introduced a very simple method, which is a combination of the COX staining and SDH staining, literally overlapping the two. In normal condition, the cytochrome oxidase prevails, and all you see is the brown stain of cytochrome oxidase. But if you have even a small decrease, a modest decrease of cytochrome oxidase, the STH stain shines through, as you see here. It's a wonderful way of revealing COX-negative fibers, even when they are not very impressive. In these pre-Raphaelite renditions of Pandora's box, we see Pandora caught in flagrante delicto, opening the box. Here it is a box, it's the English version, and letting loose all the evils. Now, in 1988, the role of Pandora was taken by Anita Harding, this wonderful neuroscientist who, among her many accomplishments, had also that of describing the first mutation, pathogenic mutation in mitochondrial DNA, which were single deletions of mitochondrial DNA in patients with myopathies, as they described in a rather general terms in their paper in Nature. And then the same year, later that year, Doug Wallace, who was then at Emory University, described the first pathogenic point mutation in a large American family with Leber's optic neuropathy, hereditary optic neuropathy, or LHON, which is, as you know, a very common, a rather common, or probably the most common cause of blindness in young adults, mostly men, mostly men. These two papers had a tremendous ripple effect, and for about the next 10 years, there was a fervor of research on mitochondrial DNA mutations. As you see represented in these two slides, this is the situation in 1988, single deletion, Anita Harding, one LHOH mutation in ND4, in the gene of subunit 4, complex 1. 2011, there are now over 200 pathogenic mutations in mitochondrial DNA, which has become very, as you see, busy with mutations. I represent them in red and blue without any political intention, only to show you that the blue ones that affect, as you see, mostly tRNA genes. I forgot to tell you earlier that mitochondrial DNA, of course, contains, besides the 13 genes encoding proteins, it contains 22 tRNA genes and 2 rRNA genes. The 2 rRNA genes are here. The 22 tRNA forms, so to speak, the punctuation between the protein coding genes. And the red mutations are in fact mutations specifically affecting protein coding genes. I actually should correct that, because I just realized that if you go to the most recent MitoMap, there are 300 mutations, pathogenic mutations. So they are still increasing. Now, a genetic classification of the mitochondrial disorders that I proposed long ago is very simple. You divide mitochondrial DNA, you divide mitochondrial diseases into those due to mutation in mitochondrial DNA and those in nuclear DNA. The fact that this list is longer is not due to the fact that there are more genes encoded by nuclear DNA, but to the fact that the mitochondrial DNA has become lazy in the course of millennia and depends now heavily on nuclear DNA for a number of essential functions. That's why this list is longer. But to go back to mitochondrial DNA, the disorders due to mitochondrial DNA can be divided into two flavors, if you want, those that affect protein synthesis in toto and those that affect single individual proteins, as I already mentioned. So let's go to the first group. The protein coding genes can be either protein synthesis can be impaired either by rearrangements of mitochondrial DNA or by mutation in tRNA genes. Now let me give you quick examples of the two situations. Oops, one slide disappeared. Let me see. Oh, here it is. Here are single deletions in mitochondrial DNA that can cause three phenotypes. Either a generalized, very severe phenotype, the so-called Kearns-Sayre syndrome, which affects essentially all tissues, but particularly the brain, the eyes, with the retinitis pigmentosa, the extraocular muscles with ptosis and ophthalmoplegia, the cerebellum, and you name it. It's a terrible disease. beginning before age 20. And here is an example of our first patient. You see how short she is, emaciated, and you may see that she has severe ptosis. She died soon after this picture was taken, but notice already an exception to the rules I already mentioned to you, the rules of mitochondrial genetics. This is her mother and this is her son, and they're both quite normal. So mitochondrial DNA single deletions are usually not inherited and not transmitted, for reasons we may discuss later. Oh, and there are two other phenotypes. One is a pure myopathy, if you want a Kienzler light, which affects only muscle, causing ptosis, ophthalmoplegia, and some degree of weakness. And then a hematological disorder, Pearson syndrome, which affects very young children, causing sideroblastic anemia and sometimes pancytopenia and pancreatic dysfunction. The next group is the defects of tRNA, and there are many of them, and I certainly don't want to review them for you. I'll use MELAS only as an example because MELAS is probably one of the most common and the most severe mitochondrial DNA-related disorders. So onset is usually in childhood. MELAS stands for mitochondrial encephalomyopathy, well, nothing special here. Lactic acidosis, again, nothing very specific. And stroke-like episode. Well, strokes you don't expect to see in children or in young adults, and the suggestion of my neurological mentor, Dr. Roland, is whenever you see a stroke in a person younger than 40, exclude MELAS. Think of MELAS and exclude it. Most cases of MELAS are due to mutation in a tRNA leucine, UUR, but while maybe 80% of cases are in fact due to this mutation, there are many other mutations, about a dozen of them, and I'll show you one very soon, that are in other mitochondrial DNA genes causing the same syndrome. Stroke typically affects the posterior cerebrum, the occipital lobes. Very often these patients have, in fact, cerebral blindness, cortical blindness. And lactic acidosis is, of course, demonstrable in the blood, but also even better by MRS in the brain. As you see, the two peaks, the doublet of lactate, both in the CSF, in the ventricles, and in the parenchyma of the brain. And notice that the mother of this affected child, completely affected child, although oligosymptomatic or maybe asymptomatic, also shows peaks of lactate. So this is a good biomarker if you are, for instance, setting up a therapeutic trial of MELAS. Okay, and let's come to the last example of mitochondrial DNA primary disorders, defects in protein coding genes. And here I have to tell you what I consider a syllogism, a mitochondrial syllogism, because we knew for a long time two disorders due to mutation in mitochondrial protein coding gene, namely Leber's optic neuropathy, I already told you about, which affects 3 subunits of complex 1, which is maternally inherited, which is multisystemic, although the retina is exquisitely more affected than other tissues. And interestingly, when you do a muscle biopsy in these patients, you never see regular fibers. The second disorder was also described by Anita Harding, I'll show you in a moment, and it's called neuropathy ataxia retinitis pigmentosa, or NARP. This is due to a mutation, or actually two potential mutations in the same site in the TPA6 gene, complex 5. It is also maternally inherited, multisystemic, and again, if you do a muscle biopsy, you do not see regular fibers. On the basis of these two disorders, we generalize, and in medicine we do generalize, we have to, until proven wrong, we generalized and concluded that all disorders due to protein coding gene mutations were maternally inherited, multisystemic, and without regular fibers in muscle. And we were wrong, and wrong. And I can attest to that because of my interest in myology, I was following and still do follow patients with recurrent myoglobinuria. And of course you see patients with defects in glycogen utilization, one important fuel, with lipid utilization, the other important fuel, that makes sense. But I ask myself, why don't we see, and for many years we didn't see, patients with respiratory chain problems. After all, that's where energy is made. If we are dealing with energy shortage, we should see them. We didn't see them because we didn't look for them. Because these numerous patients, this is only a sample, make exceptions to all three rules of mitochondrial genetics. Notice that all these patients have no family history. They are all sporadic. So these are de novo mutations. They are not multisystemic disorders, but pure myopathies with exercise intolerance and often myoglobinuria. But they all have mutations in mitochondrial DNA, either in complex 1, more often in complex 3, or in complex 4, in cytochrome oxidase. And what will the biopsy show in these patients? Here are the ragged red fibers. While I'm not showing you ragged red, I'm showing you ragged blue fibers, abundant, because this is more sensitive. But notice that they are not COX negative, they are COX positive. Why should they be COX negative? This is mutation in cytochrome B. This is complex 3 deficiency, not complex 4. So when you see a muscle biopsy that shows you, first, a mosaic pattern, which I didn't mention earlier, a mosaic pattern of ragged blue or ragged red fibers and COX positive or negative fibers, you know that you're dealing with a mitochondrial DNA disorder. The mosaic pattern is an example of heteroplasmy. We are cutting the muscle transversely, and at different section levels, the amount of mutation, the mutational load varies. So there are a lot of mutation here and here and here and here, and very few in the other fibers. But if you cut higher or lower, you would find the mutation in these fibers also. So this is heteroplasmy. Now, because there are so many mutations in mitochondrial DNA, because mitochondrial DNA is prone to spontaneous mutation, in the year after after 1988, I remember vividly how many of my fellows would come to me all excited, telling me I found another mutation in mitochondrial DNA, and you have to kind of calm them down and say, how do you know it is pathogenic? And we developed essentially canonical criteria of pathogenicity. The first two are obvious, that the mutation should not have been reported before in a normal subject, not be a polymorphism. It should alter usually an evolutionary conserved site, which means probably a functionally important site. Usually, but not always, but usually there are heteroplasmic mutation, the pathogenic one. And in this case, the mutational load should correspond more or less to the clinical severity. And then they should be functionally deleterious. But let me show one thing at a time. Heteroplasmic. Here is the importance of the mutational load. This is NARP syndrome again, described, as I told you, by Anita Harding. It causes, usually in adults with NARP, neuropathy, ataxia, retinitis pigmentosa, it has moderate mutation load, around 70%. But in the same families where NARP is present, very often there are kids with very severe neurodegenerative disease, namely Lee syndrome. And this is an example of a typical Lee syndrome, MRI, with the lesions in the basal ganglia, symmetrical lesions in the basal ganglia. And these children have much higher level of mutation, around 80 or 90%. And as I said, they often coexist in the same family. So how do we prove the functional importance of the mutation? And this was very difficult to do until Giuseppe Attardi, who actually also studied in Padua, I'm proud to say, and Michael King in 1989 described this ingenious method to show pathogenicity of a new mutation. That is, they took a permanent cell line, usually from an osteoblastoma, and this is the cell line, and they human cell line. They expose it to ethidium bromide, emptying it of its own mitochondrial DNA, creating what we call a row 0 cell line, copying from yeast genetics, and then they fuse the row 0 cell line with cell lines from a patient with the mutation that you are studying. And you created a cybrid, a cytoplasmic hybrid or a cybrid cell line, which with simple stratagems you can create with a variable amount of mutant mitochondrial DNA from 0 to 100%. And then you study these cell lines, cybrid cell lines, you study protein synthesis, you study ATP production, and you can show that in fact if in fact they are defective in these functions, and you can also establish the threshold level. At what mutation load does the function change? However, you have to, and by this method, most heteroplasmic mitochondrial DNA pathogenic mutation had high threshold and steep ones. But I have to warn you that this does not apply directly to medicine. In fact, when some of these mutations were studied in patients, the mutation does not have to have such high level to be evident functionally. So it's a warning signal. Another method to show the pathogenicity of mutation was invented by my colleague Carlos Moraes when he was with us, and that's single fiber PCR. It's a very simple method, but very effective. If you have heteroplasmy, you simply pluck, literally pluck, from a thick cross section either a COX negative fiber, like this, or succinate SDH, ragged blue and COX negative, or a normal fiber, COX positive fiber, and you do PCR analysis, and you compare the level of the mutations in the two. And you see, this is an extreme example because it's really a clean example. COX negative fibers, you see, have only mutant DNA, or sorry, I can see COX positive fibers, and the COX negative fibers have only mutant DNA, which is an extreme example. But this is a very good way of showing pathogenicity. In the year 2000, I wrote a review that I entitled Mutations in Mitochondrial DNA, Are We Scraping the Bottom of the Barrel? Which seemed to be true at the time because it looked unlikely that we would add more mutations to the ones that we knew. But in fact, there are a number of, in fact, we are not scratching the bottom of the barrel even now. And there are a number of questions and novelties. First thing is frequency, the frequency of mitochondrial DNA mutations and diseases. Are they frequent disorders? Well, finally, I'm showing you two papers from Newcastle upon Tyne where they did the best epidemiological studies. First, these disorders are more frequent than we thought. About 1 in 10,000 or 9,000 individuals, adult individuals, have mitochondrial DNA-related disorders. And then a paper came out from Patrick Chinnery, also in Newcastle, who did something that should have been done earlier, I thought. We sequenced mitochondrial DNA from over 3,000 newborns, cord blood from 3,000 subsequent newborns in Newcastle, and he looked for the 10 most common pathogenic mutations. Only 10, not the whole mitochondrial DNA. And lo and behold, he found that 1 in 200 of these children did have a pathogenic mutation. And if you consider the Mela's mutation, the 3243 mutation, 1 in 700. Now, we don't see 1 in 700 patients with Mela's mutation. And the question becomes, where are these frequent patients, these 700 patients? Either they grow happily and they live their lives without any problem because the mutation stays below the threshold in all their tissues, possible, or these patients show the mutation, or in this patient the mutation may surpass the threshold level, maybe in some tissues and not others. For example, I'd like to remind you that a study of diabetes in Japan has revealed that 3% of diabetic patients have maternal diabetes due to the Mela's mutation. So this could be true for other organs and for other disorders. I was asking myself, for example, about autism. Could a subset of autistic children have mitochondrial DNA mutation above the threshold in the brain? One thing. Another is we still find new mutations or mutations that are unusual in their phenotype. For example, this is a patient that we are about to publish, a 15-year-old girl, sporadic case, they're all sporadic, notice, who had a mutation in the gene encoding cytochrome B. Now, cytochrome B is an extremely variable gene, but it has only a relatively small number of pathogenic mutations that are all listed here. And you notice that while some are multisystemic, the majority are confined to skeletal muscle. They're the ones that cause the myopathy that I showed you before. Well, this girl had typical Miller syndrome, and yet she had a mutation convincingly pathogenic in cytochrome B. So there is still work to be done on new mutations. Haplotypes. What do we mean by haplotypes? In our migration from Africa and from our mitochondrial Eve, of course, the mitochondria have been changing, accumulating different mutations that have stabilized in different populations. And Doug Wallace, for example, proposed, and these are called haplotypes or haplogroups. Doug Wallace, for example, has proposed an interesting theory, fascinating, that certain haplotypes may cause just a slight uncoupling of phosphorylation and oxidation, and lead to production of heat instead of ATP. Some ATP becomes heat. And this may have favored the settlement of some populations in rigid climate. Very interesting. And this has been now recently supported by some functional evidence. The other interesting thing is pathogenesis. Pathogenesis is really terra incognita. After so many years, we still do not know why mitochondrial DNA mutations cause these certain diseases. Put very simply, why is Kienzler syndrome Kienzler? Why is MERV, and MELAS? After all, these are three mutations, all affecting protein synthesis in toto, in mitochondrial protein synthesis. These two affect two different tRNAs, and they are completely different. They are different. And no good clinician would ever confuse a Kienzler syndrome with a MERV or a MELAS. So why is that? One possibility would be that that mutation in different tRNAs would have different consequences. Possible, but unlikely, because you'd expect all of these mutations to cause decreased ATP production and decreased protein synthesis, and to cause, at the clinical level, kind of a swamp of symptoms, and they don't. Another possibility that was entertained by Eduardo Bonilla and Kurenai Tanji was that maybe there is a spatial distribution of the mutations, particularly in the brain, that would explain the differences between different syndromes. And they actually proven that to be true in many cases. What they did basically was immunohistochemistry and in situ hybridization. By immunohistochemistry, they compared, I hope you can see the screen better than I, but they used two antibodies, one against COX-2, which is a mitochondrial DNA encoded protein, one against noneme iron sulfur protein of complex 3, which is nuclearly encoded. And they compared the immunohistochemistry in different tissues. As you see on top, we are seeing a Kienzler patient, and we are seeing exactly the choroid plexus. And when you compare the two, you would have no doubt that there are many more mutations in the patient, that the protein, the mitochondrial DNA encoded protein is not present in the patient as it is in the control. So evident, the consequence is there are many more mutations in the patient in the choroid plexus, which is a specifically affected organ. Now, when you look at subpial vessels of the brain, Same thing in a patient with MELAS, the big difference again between the mitochondria and the protein and the nuclear encoded gene. And when you look at the olivary nuclei in MERV, again, a very specifically affected area of the brain, same difference. And when you do in situ hybridization in choroid plexus of the brain, and you use two probes, one outside the deletion and one inside the single deletion, big difference again, indicating that there is an abundance of deletion in the patient, but not in the control. So that would suggest that indeed there is a different distribution of mutation. But this begs the question of what tells the deletion to go to the, particularly to the choroid plexus, and to the MERV mutation to go to the olivary nuclei. We don't know. Now, aging is another area, well, no. Intergenomic signaling first. I began showing the same slides before to point out that while there are primary mitochondrial DNA mutations, direct hits if you want, there are also indirect hits. For the reason that I told you before, that the mitochondria became lazy in the course of evolution and now depends on the nuclear DNA for essential functions like replication, like transcription and translation. There are a group of disorders that are very interesting. They are called defect of intergenomic signaling, if you want. And this is just a scheme showing the nucleus and the mitochondrion and showing the number of functions that the nucleus controls for the mitochondria. Mitochondria really are the slave of, mitochondrial DNA is the slave of nuclear DNA. So there are a whole group of disorders, the intergenomic communication defects, that really share the genetic characteristics of Mendelian genetics and mitochondrial genetics. Let me give you some examples. Examples of multiple deletions of mitochondrial DNA. These are a series of patients with progressive sternophthalmoplegia, muscle biopsies. Look at the number of multiple deletions, while this is the example I already showed you of a patient with Kian-Zare syndrome and single deletions. And this is an example of defective replication, quantitative alteration of mitochondrial DNA. These are real patients. In fact, the original patients upon whom Carlos Moraes defined this new disorder, mitochondrial DNA depletion. When you compare the amount of nuclear DNA and mitochondrial DNA, you can see clearly that in this patient, for example, in muscle or in kidney or in other tissues, oh, this is muscle, this is liver, you see clearly that there is a defect, profound defect of mitochondrial DNA. So mitochondrial DNA replication has been impaired. And these are nuclear Mendelian disorders. Now, we thought that we had them neatly classified as defects of qualitative defect with multiple deletions, quantitative defect with depletion, and defect of translation. Well, not so fast, not so neat. In fact, in part using mitoexome sequencing in collaboration with Vamsi Muta, we found recently, and many others found, that there is no such clear differentiation. We thought the mitochondrial DNA depletion would be essentially present in infants and children, and the mitochondrial multiple deletion would be typical of adults and with their separate genetic causes. And as I show you in this slide, I hope you see there is a total admixture which is becoming more and more so between the two groups. So the same gene that can cause mitochondrial DNA depletion can also cause mitochondrial DNA multiple deletions and a different phenotype, adult usually phenotype. Why is that? Well, that becomes more understandable when we see where these genes and their proteins work. They work on the nucleotide pools of mitochondria, which is really where the building blocks of mitochondrial DNA are made. So it doesn't surprise too much that mutations in any of these genes could cause either depletion, either a defect of replication, or a defect in maintenance, multiple deletions. aging. Well, these are 3 self-portraits of Rembrandt to remind us that we are all aging. I certainly don't need reminder. And this is a study that we did many years ago, Eric Schoen particularly, looking at the common deletion, the 5,000 or so kilobases deletion in muscle. And you see, about age 40, alas, there is a tremendous increase of this mutation, of this deletion. Now, to be sure, even at this level, we are not even close to the levels of Kienzler patients. But this is not the only deletion that accumulates in muscle. There are many others. And the cumulative effect may indeed surpass the threshold effect and cause damage. It's interesting that this same age is considered the beginning of sarcopenia, the beginning of the age at which we start losing muscle mass, probably for the same reason. You know very well that there is a good theory of mitochondrial DNA involvement in aging, because mitochondrial DNA is a polyploid, mitochondrial polyploid genes, has high mutation rate, is naked, is next to the respiratory chain or next to oxidative radicals, or where they are produced, and it has a repair capacity which is fair. It's not true that it doesn't have any. It is there, but not very, very high. And nuclear DNA is the opposite, diploid, low mutation rate, protected by histones, far from the respiratory chain, and it has good repair capacity. All that makes mitochondrial DNA a very good candidate for an aging clock. And the catastrophic mitochondrial theory of aging, which was actually launched long ago by several people, postulate a vicious cycle such that somatic mutation accumulate in mitochondrial DNA, they impair OXPHOS, the impairment of oxidative phosphorylation increases the oxygen radicals, and these in turn induce more somatic mutation in mitochondrial DNA. There are alternative explanations, but this is the more common one. And this was proven, in fact, in various tissues, and induced Anita Harding with the usual wit to write a News and Views article about growing old, the most common mitochondrial disease of all. It may be. And this is an example from the Drosophila that Eduardo Bonilla studied to show again, when you do the same kind of study, immunohistochemical study that I showed you before, that mitochondrial DNA encoded proteins tend to disappear with age, while they're still intact in younger individuals. And looking at the lateral genicular body, same thing. This is the mitochondrial DNA encoded protein, nuclear encoded protein, big difference. So indeed, we do accumulate probably mutations, certainly deletions. It is more questionable whether or not point mutations accumulate with aging. Finally, and this is really my last point, are mitochondrial DNA-related diseases preventable? What do we mean by that? Well, this is a very exciting area, which is actually just starting. Yes, mitochondrial DNA, tRNA, for example, mutations like the ones causing MELAS or MERV, which are indeed rather common disorders, and indeed sisters, for example, of a patient who died of MELAS, as it happened to us, will come to you when she's in her 20s saying, I'm getting married, but I'm terrified of having a child like my sister. What can you do for me? Well, what we could do is shown here. Here you have the patient's oocyte, you do in vitro fertilization, and then you take a donor oocyte, not from a maternal relative, woman, you enucleate it, which is easy enough, and you have a cytoplasm, and you then take the pronucleus and you transfer into the host oocyte. The result will be an embryo which will have the nuclear DNA of the biological mother and father, but the mitochondrial DNA of a donor, of a healthy donor. And the child should be normal. This, in fact, has been done in monkeys. In Oregon, they've done just that. And in fact, they've perfected the technique using cells in metaphase 2 And using, this is a mito tracker, but they use a different polarized microscopy to visualize better the muscles, the nuclear spindle. Then they've taken off the spindle and created some karyoplasts. They have fused them with cytoplasm of a donor's oocyte, as I showed you before, and then do in vitro fertilization, implantation, and the result was beautiful. Two twin pups called Mito and Tracker, who were shown to have, in fact, almost no mitochondrial DNA from the mother, from the biological mother, but all from the donor. And the nuclear DNA was, of course, maternal. This is now happening. Studies are now occurring for humans. This has been done in Newcastle upon Tyne. This shows just the two possibilities I showed you before. You can either have a fertilized egg and do the procedure that I showed you before, or do the procedure later. I mean, it doesn't matter. The procedure is basically the same, and you have then to follow, of course, the development of the fetus. Now, that has still a lot of ethical objections, although I frankly do not know why there is such terrible objection. I must say that at Columbia we have now applied to do the same kind of research. We also have an excellent collaboration with our in vitro fertilization clinic. And I don't know why, because this same technique was applied, but partially, in the so-called rejuvenation procedure that was used in many IVF clinics, where an older woman would want to have IVF, and a portion of the cytoplasm of her oocyte was removed, let's say 25–30%, and replaced with the cytoplasm of a younger egg, and the results apparently were better. The rate of success increased. What's the difference? Those babies were also heteroplasmic babies, even more, in fact, than this would be. But there is objection, and even in England, when this study started, a tabloid appeared with this lurid title. So we are up for a battle. This is a slide recognizing all my, this is the way I feel as a good New Yorker, but here is a big orchestra, and the one who takes the applause at the end is the guy who plays the Tinkerbells. And that's the way I feel, while the work is done by a lot of collaborators. I apologize for the fact that the young ones did not fit in here. These are my more traditional collaborators, particularly Eric Schoen and Micio Irano, with whom I really continue to collaborate with great joy, and Daryl DeVivo and so on. Petra Kaufman, who left us, unfortunately, to come to this place. and the support of NICHD and NINDS, and also the Mario Mitochondrial Disease Foundation, but particularly NIHD and NINDS, because they recently made possible a dream of mine, the creation of a consortium, of a North American mitochondrial disease consortium, that now has this number of collaborators. There will be more centers, certainly more adjoining, where we will try to help patients reach a correct diagnosis, help colleagues make correct diagnosis, and do biological histories of different diseases, and guide therapeutic trials. So this is a tremendous, I'm grateful for that. And finally, as a good Italian, because today is March 21st, have a happy spring. And thank you very much for your attention.
We have time for a few questions.
So, So in population genetics, deleterious mutations usually don't accumulate to very high numbers. And in fact, mitochondrial mutations through generations are not accumulating catastrophic mutations. So I wonder how in an individual then, an individual mutation in a mitochondria can take over and achieve a level above threshold.
Well, that's an interesting question. For example, for what regards deletions, it looks like the deletions in fact have an advantage. For example, children, the example I can give you is also an example of mitotic segregation. Children with Pearson syndrome, if you treat them with frequent, well, usually die within a year, but if you treat them with transfusions, some of them survive and go through a well period, only tragically to develop Kearns-Sayre syndrome later. It has been shown very clearly that while the deletion, in that case single deletion, is eliminated by the rapidly dividing cells of the bone marrow, and that's why their original disease disappears, in post-mitotic tissues, in fact, the mutation increases, as if it had an advantage, which in fact it seems to have, and reaches, after a few years, the threshold level and causes Kearns-Sayre syndrome. So it is a strange and counterintuitive phenomenon, but it has been observed and documented very clearly. Now, somebody may say, I was discussing with somebody today, that because the deletion, of course, is short, is a big deletion makes the mitochondrial DNA smaller, replication will be faster. But in an initial experiment, at least done by Carlos Moraes and Eric Schoen, they couldn't prove that. But there may be other experiments that did show that. That may be one element, one advantage.
So I have actually a clinical question. I see a fair number of patients with suspected mitochondrial diseases here in the neurogenetics clinic, and one of the things that we run into frequently, it's we see a patient that we suspect has a mitochondrial disease clinically, and of course we try to look at the mitochondrial DNA, and we more often than not get a change that when we try to look it up in the databases, it's not reported as a polymorphism. It hasn't necessarily been reported as pathogenic, and now as the databases get more complex, in particular I'm thinking of the Leiden database, we started to notice that we're seeing patients that have groups of 2 or 3 mutations in different genes in the mitochondrial DNA that are being reported together in high degree of heteroplasmy, and I'm talking about more than 99%. So my question to you clinically is, what do you do with those patients, and do you have any advice, or should we just wait a few more years?
If I understand correctly, you have patients where you find not one mutation, but more than one, 2 or 3
yes.
heteroplasmic mutations in mitochondrial DNA?
Yes.
Which have
they're not reported as SNPs.
Which have not been previously reported.
And they have not been reported as pathogenic.
Well,
but they have been reported in these databases. So someone somewhere, these are like the Leiden database is an open database that if you're interested in patient, you can just put it there. So obviously other people are seeing the same group of mutations together, yet it's not clear if they're pathogenic yet or not.
It's very interesting. As far as I know, in a few cases that I know from the literature, when this has happened, really one mutation was pathogenic and the others were shown not to be pathogenic. But you cannot exclude, of course, the simultaneous presence and the synergy of two mutations or 3 mutations. But that's a very interesting and unusual phenomenon, which I think you should, if you can document clearly that all 3 mutations are pathogenic or are synergistic, you should report. It's most unusual.
So thank you very much.
Thank you.
Please join us in the back for our FAES cookie and coffee break. Oh, it's in the library. I'm sorry. In the library.
Thank you.